Hi, I'm here today at the Louisiana University Marine Consortium, or LUMCOM, the main center for marine research in Louisiana. Hi, my name is Dr. Kelly Robinson. I'm a marine ecologist who specializes in jellyfish. There's been some questions about whether jelly populations are increasing. But to know the answer to this question, we first need to collect some data. We're going to go out today on the Arvea acadiana to collect data to see if we can understand why these jelly populations may be changing in size. One of the ways that we can sample jellyfish is dip netting, where we take them out of the ocean one at a time. So the first step that you want to do is actually get a bucket that's half full of water. Throw a 5-gallon bucket over the side that's tied off to the line and just grab some water from the surface. The reason that you need to get a bucket of water is that you want to have basically a volume of water that the jelly can be put in as soon as it comes out of the ocean. And then the next step is you're going to have 2 people, and one of them is a spotter and the other one holds the net. The spotter's job is to look for jellyfish that are at the surface, and so they keep their eye out, and as soon as they spot one, they point to it like a man overboard drill, and they just keep pointing at it. And the person who has the dip net then focuses on that point in the water and tracks the jelly. And so in coordination with the boat captain, who tries to drive as close as possible to it, the dip netter then scoops the jelly up out of the water and puts it into the bucket that you've already filled with water. The reason we want to sample jellyfish by dip netting is that it's the least damaging sampling method. It allows you to be very gentle, and so we don't damage jellyfish, which are very fragile organisms. They're made of 90 to 95% water, so they're very easily torn apart. And so the dip netting method allows us to collect live animals to bring back to the laboratory. So collecting data from one place is not enough to understand changes at the ecosystem level. So to do that, we use a technique called trawling to collect jellies from many different places. We go around to different places around our ecosystem and we put a trawl in, and then we bring the trawl up and we count the number of jellyfish that are in that trawl. So trawling is a technique that allows us to capture the number and the distribution of jellyfish in an instant, in a snapshot. But if we want to understand how jellyfish populations are changing through time, whether they're increasing or decreasing, we need to have repeated samplings. And to do this, one of the techniques that we use is to take a set of sampling sites or areas where we trawl and go back to these sites repeatedly over time. And what this allows us to do is develop a time series that then can tell us whether jellyfish in the ecosystem are increasing or decreasing. And the reason through this is that if we have these time series, then we can understand how different environmental factors, whether it be temperature or salinity or oxygen or nutrient runoff, may be affecting these different components. Put these things that were not interested back into the ocean, and we can then use measuring tools to then count and then take measurements of the jellyfish. So my work as a marine ecologist consists of 3 main parts. I go out on the field, on the boat, and I sample organisms in the ecosystem. I also bring them back in the lab to do experiments, and then I take what I learned there and I put them into ecosystem models. Scientists need help, not only from citizens, to let us know when you see phytoplankton blooms or when you see jellyfish off your docks, but also we just need more scientists, because the more people that we have monitoring our oceans, the more information we'll have and the closer and more quickly we can get to answers.